That was nice flying, Kylie.
Thank you. Feel strong. I feel
It's
good.
actually like dramatic improvements every time you get on. It's really
Thank
impressive.
you. Thank you.
It's all fantastic. It's just more fantastic.
Exactly.
Thank you.
All right.
I like criticism, but soft criticism.
We'll leave that to Mike.
Yes, I'll take it.
All right, I'm going to start taking steps up here. Bridge nav. Request 20-meter step bearing 140.
No, you got some really good shots just now. It's good.
And we'll have about 10 to 15 minutes on this one before we change over.
Right on time. I am getting a wicked headache.
Chris, your dog has another question.
Oh, yeah.
It's not for you, though.
Oh.
It's for the ROV team. And Kelsey's wondering if the ROVs have a temperature range that they operate in or cannot operate in.
They do, I guess. They have a very wide range that they can operate in, but they're only, the hydraulic system is only cooled by the ambient water. So we do run into problems if we have to stay on the surface in a very hot place, like such as we are here. They'll overheat very quickly. Beyond that, though, not really. You can put them in any ocean temperature that we have. It's just if it's too hot is an issue.
So we don't tend to dive shallow for that reason.
That's right,
In the
yeah.
tropics.
Bridge nav. One more step, please.
We're listening to the bridge.
Yeah.
I was like, we heard it, we're good. He was like, no you ain't.
If you guys hear Wayne talking to me on SBL,
No.
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 Right.
What's that thing?
What's this thing? Zoom in there, video.
Bridge nav.
Is it like
Looks like
a
a
sponge deposit?
Yeah, the glass sponge.
Yeah, one of those dark brown glass
One more step, please.
sponges.
Thank you. The rare furry rock.
I know sometimes when sponges get sediment covered, it's because they're deceased and no longer filtering the water. Yeah, Steve just confirmed that it's a dead bundle of sponge fibers, he says, so it might be partially collapsed sponge as well.
Looks like a certain president's hairpiece. Just on display.
So another biology question for you, Jamie. When we come across things that we're not quite sure what they are, how do we determine whether or not it's a new species, and do we have to sample it to tell? What's that process?
That's a great question. So when we see something that we don't recognize or we think might be a new species, we can't get very far with new species identification solely from pictures. So you would need a sample so that genetic analyses could be run and closer morphological analyses can be done than what can be determined just from a photo. And that process to fully determine if it's a new species or not can take several years of bench work and comparing to other related species genetically and morphologically. So yeah, it can be a multi-year process to ultimately determine if something we saw was a new species or not.
Changing ship's heading ever so slightly.
Roger.
This reminds me of a roller coaster.
Yeah, pretty neat one.
Bridge nav. One more step, please.
Some fish to the right.
Oh yeah, we missed him.
Looks like another grenadier or rat tail of some sort.
Zoom in there.
It's got something living on it. Or is that, yeah.
Probably a isopod.
Mm-hmm.
Nice zoom.
Coming in a little tighter.
Yep.
Full zoom.
That's cool seeing the individual rays on the fin.
Yeah.
That's cool. See, this one's not shy at all.
Okay, I'll have to move on.
Reset DVL real quick.
Roger.
All set. Bridge nav. One more step, please. Thank you.
So all these features seem to be pretty similar geologically.
Yeah, definitely. Kind of thin
Mm-hmm.
sheet flows, and then they'll get thicker as we get more towards the top.
We got a question coming about how long sponges and corals can live. Deep sea corals can live on the scale of hundreds to even thousands of years. I think up to 4,000 years was the oldest one that's been observed so far. So quite a long time. And sponges in the deep sea can live also on the range of hundreds of years, not as long as corals. We don't think 200 years would be pretty long, could go a bit older than that. In contrast, shallow water sponges only live a few years, so the deep sea and the colder temperatures allows some animals to live a lot longer than their shallow water counterparts.
Do you know where the 4,000-year-old coral was?
No, it was one that
Bridge
Steve
nav.
mentioned. I don't know.
In the Gulf?
I don't know where it was observed.
One more step, please.
Steve is on Science Chat though, so maybe he can let us know.
Also curious how you know it's 4,000 years old. How do you date a coral?
You have to sample it, and you can look at growth rings, sort of like a tree.
Okay. That makes sense. It's a lot of rings to count.
Can you tell things about the environment, like over those 4,000 years from sampling the biology of a coral like that? Like the way that they do with like sampling, I don't know, I'll say like carbon that's in cores that they get from deep sea?
But you definitely can with some coal, because fossil coals
Zoom
have
in
been
here.
drilled, and you can take changes in ocean chemistry through time by analyzing what the chemistry they kind of built this basis out of.
Right.
I don't know about deep sea coals. I'm assuming the same, but less sampled.
Yeah, the base is really helpful when you're trying to age corals.
Right, okay. You can see what was going on when it started, at least.
I don't know about corals, but I know in terms of the rings and growth with trees, and they do the same thing with scales and fish. Depending on the amount of growth between
Okay, come on.
seasons, you can get a rough estimate on at least how favorable conditions were for them, and then estimate it based on the conditions.
Oh, can we hold position? Steve would like to sample that coral we just zoomed in on.
Sure thing.
Yeah, you want me to
Yeah,
hold the ship?
hold the ship up.
Roger that. Bridge nav.
All right, biosample.
Of course, we hold position.
And
Thank
his
you.
preferred sample method is to cut it off and then slurp it into one of the available chambers.
Roger. We're doing a shift change. It's just coming in the door. I think I'll probably hand it off to them.
Okay.
Let's see. Oh, there it is. Open the iris up a little bit. Morning. Setting you up for a sample here.
Alrighty.
Yep.
Morning.
Good morning.
Good morning.
Someone else is attached.
So do we want to slurp this guy?
That's the plan, I think. I want to slurp this guy.
Okay.
I might just grab it and move till I'm in a better spot. That might be what I'll do. Try and do that in a hurry.
I'm backing up towards you.
Roger. How much do you want?
I think just a small piece.
Roger.
He
Zoom
said.
in a little bit, please.
Yeah, like 10 to 15 centimeters.
Yeah.
How about right here?
I think that's good.
Yeah. It's good there? Okay.
Yeah. That's more than enough.
Okay, come wide, please.
Come wide.
Okay. Just gonna hold it out here like a party favor in the meantime, and you can start coming up on your winch, and I'll come towards you a little bit.
This is a primnoid coral, Steve?
Must be eating.
Good.
Yep. Megan is doing a show, interaction show thing. Your mic's hot. Okay, I think I'm safe enough here. I can do some slurp action. Which jar would you like this in, Zuna?
I'm looking. So 2, 3, 4, or 5 are good.
Okay. Could you please line up that with 2, 3, 4, or 5, and then after that, give me bubble on front porch, please. Perfect. That's 5. And yeah, bubble on front porch, please. Oh, or sorry, I said that. I don't mean that. Can you give me on the slurp hose? Just another perspective there.
Good morning, 8 to 12.
Good morning, Wayne. Okay, let's start with 30% suction, please. And Wayne, could you zoom in about halfway, please?
Halfway zoom.
That's good there. Okay, maybe let's bump it up to 50. Thread the needle. There we go. That'll do it. Okay, come wide, please.
Coming wide.
And when you see that in there, you can kill the suction.
Wide. Full
Just
wide.
go in there.
I haven't seen it yet.
Oh, there you go.
Yep. Is it in 5?
Yeah, you can kill the suction now.
Yeah, it's
That's
between
correct.
4
It's
and
in
6.
5. Right between 4 and 6. 4.5. And you can flush whenever. Cool. Sample taken. No, I can't.
What is in Bio 7?
I don't know, but it looks pretty cool.
Yes, it does.
Wayne, you could give both Zeuses a little bump.
Okay.
All right, I'm coming up. Coming underneath you.
Yeah. Sure.
Look at this noise. Now that's a new one. That's cool. I was just thinking that. Thank you. Okay, it's good.
Okay, that's good to know. That's fine. No, I just asked Steve if we, that icicle coral thing we saw last night, I'm not sure if they got any samples of that or
What's our
if they
bearing?
want it. I just asked them.
143.
4-3. Thank you.
Yeah. Okay.
In theory.
That daisy chain looks really good. There's the one long one at the start, then everything else looks rock solid.
Yeah, who did that?
I wonder.
We'll never know.
I think the initials are TS.
TS or ST?
Tom Sawyer.
I'm sorry, was that BS?
Wait, could you iris a little bit, please?
Yes.
Reach nav.
Thanks.
Another step.
Yeah, what is it in 7?
Did you notice what the oil temp was on the winch?
No.
Okay. Just out of curiosity. Was it really hot down there?
No.
Oh, cool. Why don't we have a little zoom on this puppy here?
There's puppies down here?
Well, this puppy, yeah. It's a sponge puppy.
Yeah, guys.
Sponge?
Ooh.
We really did slurp a Pelagothuria in 7.
Oh, they did. Good.
Yeah, they were looking
Okay.
for a
'Cause I did.
pelagic holothurian.
Good.
Apparently they slurped one.
Yeah, because I saw one on the last dive that they didn't slurp, and I was kind of surprised they didn't go after it. But
All
at
right,
least
come
they got
on.
one. That's
Come
good.
on.
Bridge, nav.
Crinoid.
What's this one?
Is that a crinoid?
Is it a crinoid? Go ahead and
I
zoom
don't
in there,
know.
please.
No, it looks like a.
Is that a mushroom on a stalk?
It looks like
Oh,
a stock
wow,
mushroom, yeah.
2 stops?
That's
2 more
interesting.
steps, sorry.
I can actually see into the. That's interesting.
That's a really neat looking one. I don't think I've seen that before.
He's a little bit different.
Oh.
We're all a little bit different way.
That's what makes us wonderful.
Okay. Come wide.
All right.
Something off to the right there. Mid-screen.
Off to the right. I am
Yeah,
seeing
just
nothing.
to the right of the eyes, or the lasers, a little bit. Right.
Oh, I see. Just
Yeah, just to the
Directly
right
to the
now.
right.
Just now to the northeast of it there. Yeah.
Oh, I see it.
go ahead and zoom.
Zooming in.
Wow, good eye. Huh?
It's the caffeine.
I still don't see it.
I still don't either.
There's a little bit something there. I thought there was.
Up, down, left, right.
No, maybe it was the caffeine.
The caffeine.
Yeah.
Okay.
All right, come
Coming
on.
wide.
Rob
Seeing
can see caffeine.
Wild caffeine.
There's a little something there.
All right, you bump into wide.
Wide.
Thanks.
So, Trevor, have you seen a satisfactory predation event?
A which predation event?
A satisfactory one. That's what you were looking for.
It is not. I think that was Miles looking for that.
Was it?
That was me.
Trevor Miles.
Oh.
Not in a predation event.
Have
Not a satisfactory one, anyway.
Right. Starfish eating coral is not
Oh.
satisfactory.
Could you classify different predation events based on their length of time it takes to happen? Like, you know, a shark eating a seal is going to be pretty fast, whereas like a sea star eating a coral is pretty slow. Is there different classifications for that? Like, I don't know.
I'm sure we could make something up.
We could absolutely
The
speculate
aggregate
wildly. And I'm good at that.
There's no scientific known scale to measure gluttony
To
that
measure
I know of.
gluttony.
On the gluttony scale, that's a 2. guess we can make it up.
Yeah.
There's an urchin, or no,
Anemone?
anemone. It's a dark one.
Oh, coral on the overhang there.
Bridge now.
Can we zoom on the anemone?
Yes, we can. Go ahead, Wayne.
Okay. I'll wait
more steps?
for. There we go.
What do you think, Steve? What is it? Was it Steve that asked for that? I think it was Steve that asked for that.
Yeah, I think so.
The mystery voice from afar.
Yeah, this is a ceryanthid anemone, tube, or sometimes they're tube anemones, but they have 2 rows of tentacles, which distinguishes them.
Cool. All right, come wide. I'll move along.
Yeah, it seems like there's not as much coral here as there was last night on the last peak. Fish.
It is familiar.
I think we'll wait for coffee to kick in.
Rage Nav. Two more
Before what?
sips.
Introductions.
Introductions? Who said that?
Somebody asked for introductions.
Oh, I didn't hear that.
You're mistaken, Wayne.
I'm hearing things.
I think so.
Sponge to the right.
Sorry, you see something fun there?
Oh.
Sponge lower right. Let's check it out.
Right.
Okay, go ahead and zoom there, please.
Okay. Zooming in.
I'm going to mess with your camera a sec.
Oh. Oh, oh, oh.
All right, now we're good.
Oh.
Tunicate? Steve says it's a carnivorous tunicate.
Carnivorous tunicate.
Okay, coming
All right,
wide.
come wide.
No.
So, did I hear that we have a defined recovery time now?
Yeah, it looks like we're going to be off bottom at 10:30 today.
Off bottom at 10:30.
Yep.
Roger that.
So we'll just go to the top of this peak. Let's grab a sample, then just proceed along these peaks to see what we can get for a potential slurp of biology.
Great. Sounds good.
Do we want another rock sample?
Yeah, the rock sample at the top. There's a little coral right there. Just to the left
Zoom
of
in on
the
the
eyes.
coral.
Zooming in.
Nice yellow one.
Yep.
Do we want to bounce off this peak once we get there and try and make the next one, or
Yeah, we could do that.
do you want to try and
I
back
don't
down
want to do
again?
a moonwalk again.
Come on. No, I'm okay with not doing that too. All right, come wide, please.
Coming wide.
It was a great experience.
No.
I mean, typically I try to do everything twice just to make sure the first time wasn't a mistake.
Even mistakes?
Yeah. And there's one on the right there, a little purple one for Wayne.
Oh yeah. Purple Wayne coral.
2 more steps.
Peak 2.0. What was peak 1.0? Last night's?
No.
Another stalk mushroom
See
coral.
if there is a target.
Just peak. Ah.
Oh, there's a big one to the right.
Oh, there's a good
Fan.
one. Oops, I need to turn that down a bit.
All right.
I think this one was.
Okay, go ahead and zoom there, Wayne.
Not sure though.
Is that the
I'm gonna keep
candelabra
approaching
one?
it.
Yeah, it's a bamboo. Yeah, nice.
Target peak, peak, and peak 2.0.
Nice.
Solid naming convention.
Yeah.
Little squat.
Yeah. That's bamboo, is that true?
Yep.
Yep.
Want to say yeah.
The divisions in the stock.
Right, that's what I was looking at, yeah. It's cool. Beautiful animals.
Two of them.
Wow.
They don't get along.
You stay at that end, I'll stay at this end. All right, come wide.
Come wide.
I got a mosey for a sec here.
Look, there's another one right there.
That's a,
Is that the icicle one?
yeah, one of the larger icicle
Yeah.
ones.
A little fish there. Some of the yellow corals there.
It's really easy to fall behind.
I'm moving too fast for you.
Nope, no, you're good. I just get distracted by pretty science.
Yellow corals. Yeah, they're much more up these pillows here
Let's
compared
take
to
a look at
the
this one.
sheets last night. Yeah, here's a nice little.
Okay.
Is that a
Is
stalk
this
sponge?
stock sponge? Yeah.
Bridge nav. One
Go ahead,
more
zoom in
step.
the stock sponge, Wayne. We gotta do that.
Cool.
Aye, aye.
Huh.
shrimpy in the back there, it looks like. Oop. Ooh, sorry,
He's
bouncy a little bit.
Sticking his
Oh,
head
he's
out.
gone.
Okay. He's
Kind of looks like a toadstool.
Oh, that's crazy.
hanging out.
All right, come wide, I'll move along.
Coming wide. Wide.
Thank you.
Yeah, Steve says the stock glass sponge is in the genus Calaphicus. A little more of the yellow corals here. An old sponge base up there right in front of us.
Oh yeah.
Another sponge off in the distance.
How can you tell the difference between a sponge base and a coral base from this distance?
It just looks like it has that filamentous look to it.
Oh, okay. Nice looking sponge.
Yeah. Yeah.
Spiny.
Ooh, he's full of stars.
Yeah. Starry-eyed. We can zoom in on that one. We got time.
Yeah, that's nice.
Looks like they're picking them
Wow.
apart.
Oh.
Yeah.
Is it a sponge?
It was a sponge.
Quite a
It's
lattice
thick.
Yeah.
work on it.
He's collapsed at the tip.
Yeah, that's cool. I don't think I've seen a sponge like that before. Oop, there's the ground. Let's go back this way.
Nice. Steve, that's an old euplectical sponge. No, he said he's wrong. He was just mistaken before. Walteria species.
All right, come wide.
Coming wide. Wide.
Ship is holding.
What are those
Roger that.
yellow corals, Steve?
Yeah.
They're everywhere. Oh, thanks. It says it's Plexauridae.
I sense a peak.
Yep. Time for a rock. Well, very little sedimentation up here.
False summit.
What do you think the layback's like there, Miles? You think if I go to this little rock field up here, I'll be safe?
Maybe the far end of it. If that
Okay.
was a false summit. There's still a little bit more to go.
Roger that.
If on. Oh.
Rob, are you looking at the chat?
Okay. That. Okay.
Any specific style of rock? Big, small, angly, round?
Yeah, I mean, it's the angle if you have, it's usually a little better.
Whatever we get, we have to be very careful describing it because we're going to put it with other rocks.
Uh-oh.
Yeah.
A distinctive rock.
Yeah.
Yeah.
Steve, roger that, no problem.
The
purple one
would be good.
Yeah, exactly. I might go right here. This seems like an okay spot, eh?
Looks like there's plenty of...
Are we looking for a smaller rock then? Because we're going to be jamming it in with something else?
Probably. I don't really know how big the rocks in the boxes are.
I also remember the one in the starboard forward was kind of big, but that's where the other cookie cutter starfish went.
Yeah, I think we're gonna go forward bio box.
Port side?
Port section, yeah.
Okay. I got a rock in mind. Try and nestle in here and grab it.
Oh, plenty.
Yeah, actually, see that purple coral
Full
there?
wide.
Can we actually get that one?
Purple coral. Oh
Yeah,
yeah, let's
see.
have a quick zoom on that first off. Go ahead, Wayne.
Yep. Okay, zooming in.
Yeah.
That looks quite big.
Yeah,
The coral or the rock?
the rock.
The rock.
I can poke it and see if
Can
it's...
you shoot your
let's see.
lasers on it?
Yep, you got to zoom out then.
Yeah, it's not so big.
It might be big if there's already a rock in the box.
Well, I think it's worth a try.
Okay, come full wide, please.
Full wide.
What am I doing? I'm going to do this.
I'm gonna look in the box first.
We could look in the box first. That's not a bad
Yeah.
idea. Yeah, okay, go ahead and open that. It's the tool tray.
Do you know what the purple coral is, Steve? I think he's on his way up to the van.
There's 2 rocks in there already. Wow.
Well, this one I think is unique enough if the coral's on
Victor
it.
Gorge. Oh, that's the one we were actually
All right,
looking
you
for.
can close that again so the sea star doesn't escape. What do you think? Is that purple guy going to fit on top of the other 2?
Maybe go...
I think we can try it.
Yeah, let's just try it. And if
Okay,
the-
Wayne, could you push in, please?
Pushing in.
About halfway, I'd be fine. That's good there.
Go.
And we're working on the assumption that it's loose, which we don't know yet.
Oh yeah, true.
Let's just poke it and see.
Boop.
Rejected.
Looks like...
Eh.
That... No, all the other rocks moved.
Yeah, it's a little bigger than you think.
But that one on top, it was loose. If that's sufficient.
It is moving, but it's not moving a lot.
Yeah.
It's really big.
There you go.
Yeah.
Oh, it's quite big. Huh?
Yeah, I don't think that's going to go in the box.
No.
Well,
We can pick it up and try, though. Always game to try stuff.
Coming.
Let's keep it zoomed in there for a sec. Just
Okay.
put the